Okay, so today's video is going to talk about active transport. And last time we talked about passive or diffusion. And so this time we're talking about active transport, which is going to be basically the opposite of passive transport or diffusion. So if it's the opposite, then this is going to require energy. It's going to require an input of ATP, where diffusion or passive transport did not. So osmosis, simple diffusion, facilitated diffusion, none of those require ATP. Active transport, however, will require ATP. It's going to require energy, ATP made by that mitochondria. So that means we're going this time from a low concentration to a high concentration. So essentially we're going against what's called the concentration gradient. Okay, so we're going against the gradient. We're going from an area where there's a lot of something. We're going from an area where there's a little bit of something. You can see that up here in this example. You look at the little orange hexagons. You have a low concentration, okay, of sodium here, and you're trying to force it back where you already have a high concentration of sodium. So it requires ATP or energy. It goes from low concentration to high, and it goes against the concentration gradient. If you think back to the example with the river, okay, so we've got some people that float down the river. They just lay on their back and they just float down the river, no problem. Okay, that's our simple diffusion, our passive diffusion. They're going with the concentration gradient, so they're going with the current, with the concentration gradient. They're going from high to low, and that doesn't require any energy just to lay there. Some people, they try to float on their back and they sink like a stone. And so they need a float, they need a raft, they need an inner tube. And that, once they're sitting on the raft or they're sitting on the inner tube, they can still float down the river. They can still go from high to low. They can still go with the current, with the concentration gradient. And that doesn't require any energy to sit in that inner tube. So that's our facilitated diffusion. They need a little help, but once they have that little help, they're good to go. Active transport is when you have to turn around and swim back up the river. That requires energy. It requires ATP. You're going from low to high, and you're going against the gradient. You're going against the current. You're swimming up the river. Okay, so that's going to be our active transport. We're going to look at a couple of different examples of active transport and why a cell would even need to do that. We're going to look first at what's called endocytosis. Endo meaning inside. Cyto is the cell. So in this case, we're taking things into the cell. You can see the simplified picture up here of endocytosis, taking in that pink molecule there. Okay, but when the cell does endocytosis, okay, it forms a vesicle. Remember, a vesicle is that little storage sac. Okay, so it forms a vesicle around whatever it is that it's trying to take into the cell, and it's going to encircle whatever it is, and it's going to pinch that vesicle off, and now that object is going to be inside of the cell. Okay, so your vesicle membrane is going to be the same as your cell membrane because they were made of the same stuff. Let's take a look at the picture. Okay, so we've got two basic kinds of endocytosis. We have what's called phagocytosis. Phago means to eat. Cyto is cell. Okay, so we've got what's called cell eating. And in this instance, with phagocytosis, you're taking in a much larger particle. You can see phagocytosis happening here in this picture. There is a larger particle out there that the cell is basically going to reach out and grab and bring it back into. It's looking for something that's specific usually. Okay, so phagocytosis can be specific to a particular particle. Things that would do this would be, for an example, an amoeba. I'll show you a picture of one in just a minute. Okay, so an amoeba is a unicellular organism, so it's a one-celled organism, and this is how it eats. It'll reach out and it will engulf particles and bring them back in. In your body, your white blood cells, your macrophages, okay, this is how they will eat the foreign particles that are in your cell. They'll basically reach out, encircle them, and bring them back in and digest them and break them down. So here you can see the amoeba in the top left corner here doing the phagocytosis, the cell eating. There's its food particle here, and it reaches out, okay, it engulfs it, forms in this case, a vacuole because it's larger, pinches that in, okay. There's lysosomes then that attach to the vacuole. Remember, lysosomes digest things, and so it's going to digest or break that food down so it can use the nutrients from that food. The other type of endocytosis is what's called Pinocytosis. You can see that in your middle picture down at the bottom. Okay, this one right here is Pinocytosis. This is sometimes called cell drinking. The last one was cell eating. This is sometimes called cell drinking. This is not as specific as phagocytosis can be. Remember, phagocytosis, it's looking for a particular piece of food, it's looking for a particular foreign particle, your white blood cells that are in your body, and they're looking for something that's invading. Pinocytosis, it just forms a little vesicle. So you see it's got this indention here in the cell membrane, and whatever happened to fall in there is what fell in there. So it's non-specific. It's whatever's in the solution outside of the cell is what's going to come into the cell. And it will, but just like phagocytosis, it'll pinch that off, form the little vesicle, and then the cell can use whatever the contents are of it. The opposite of this is what's going to be called exocytosis. So your exocytosis, exo being outside, cyto is cell. So exocytosis is for secretions, releasing things outside of the cell. You're secreting or getting rid of, okay? You're releasing things outside of the cell. And so when a cell, let's say you're talking about your body and you've got a cell that produces hormones, those hormones have to be secreted from the cell. So your body made these little green hormones, and now this vesicle fuses with your cell membrane. So you can see it joins with your cell membrane here, and it opens up and releases those hormones. It secretes those hormones out into your bloodstream so they can go where they need to go. And then that vesicle, see, just becomes part of the cell membrane. And so the vesicle and the cell membrane are made of the same thing. You can see in your picture over on the right, okay, you can see both of these things happening here. Okay, so you can see you've got exocytosis happening. Okay, it's going to release these out. Okay, and then you also have endocytosis. It's taking these particles into the cell. So like I said, the two are essentially opposites, but both of those are going to require energy. So endocytosis and exocytosis are both going to be types of active transport. Our last example of active transport that we're going to look at is a pump. It's an example of a pump. This one is the sodium potassium pump. And you will never see it written out like this. You will always see it abbreviated, your sodium potassium pump. Okay, and so it's a pump. So obvious, to me that implies that it's going to be active. It is working, it requires energy, it's moving things from a low to a high concentration and against their concentration gradient. This sodium potassium pump is really important in nerve conduction. So sending messages throughout your body, making sure your nerves work properly. Once the message has been transmitted, your nerves need to reset themselves. And so they will use this sodium potassium pump to reset themselves. You'll see if you look at your concentrations, you have a high concentration of sodium on the outside and a low concentration on the inside. And then you have a high concentration of potassium on the inside and a low on the outside. And what is happening with these is sodium is getting pumped out and potassium is getting pumped in. And so you're creating on purpose a difference where you're going to have lots of sodium on the outside and lots of potassium on the inside, which, like I said, is very important for nerve conduction. So this is another type of active transport. And so you need to be able to tell the difference between active versus passive transport.